My name is Chad MacArthur, and I'm a staff scientist at Thermo Fisher Scientific. Today, I'd like to talk to you about a white paper my colleagues recently produced in collaboration with Benjamin Fryer, PhD at Pluripotent Incorporated. In this white paper, we specifically focus on the how and why of integrating high-quality raw materials and ancillary materials into the manufacturing process of pluripotent stem cell therapies throughout the product development process. Unlike many other forms of cell therapeutics that are tailored for individual patients and rely on small batch production, allogeneic pluripotent stem cell therapies require the establishment of a master cell bank that must last the lifetime of the product. Therefore, raw material quality is paramount from the beginning of the development process. We review the various factors that can have an impact on the cost, speed, safety, and effectiveness of psc-derived therapies from the standpoint of FDA regulation, process development, master cell banking, release testing, product lifecycle planning, and the role of suppliers. Taking these factors into consideration, early and vigilant planning can help companies and academic centers developing such therapies avoid costly mistakes and increase the chances of regulatory approval and commercial success. We provide an overview of the approval process for clinical-grade cell therapy products, beginning from the initial stages of generation of non-GMP iPSC lines to initial meetings with regulatory agencies such as the FDA, development of GMP master cell banks, working cell banks, documentation, testing, and process development, through the final BLA submission and ultimate approval of the cell therapy product. I invite you to read the full white paper to gather further insights into manufacturing pluripotent stem cell therapeutics.